Hi, I'm Katie.
And my name's Kyler.
And today we will be talking about clock gene expression via melatonin 1 receptors.
So to get this whole thing started, we need melatonin. Melatonin is produced in the pineal gland. The way this works is it takes tryptophan in the daytime and converts it to serotonin. Then, in the nighttime, it receives norepinephrine from the suprachiasmatic nucleus up the pineal, which tells it to turn that serotonin into the melatonin. This whole process is regulated right here by the retinal response to light. So our eyes sense the ambient light and send a message down photoreceptive ganglion cells using the retinohypothalamic tract to the suprachiasmatic nucleus. There, the suprachiasmatic nucleus interprets this message, and it uses norepinephrine to tell the pineal gland whether to inhibit or stimulate the production of melatonin. Once we have melatonin, it travels around via either albumin in the bloodstream or by cerebral spinal fluid. Once it has traveled around, it's reached its target, it can go to the cells and actually get to its receptor.
And which receptor is that? Of course, the melatonin 1 receptor is the one we're looking at, which is a G protein coupled receptor, or a GPCR. And you can tell by this diagram, it has the 7 alpha helices that are transmembrane, forming extracellular loops and intracellular loops. And then it also has some B-pleated sheets folding on the outside and the inside as well. On the outside, we have the N-terminal, which just means the amine group is out that way. The carboxyl group, the C-terminal, is on the intracellular side. And the ligand pocket, where the actual signal will be received, is between the 6th, the 7th, and the 5th helices. And over this little pocket, the second extracellular loop, E2, kind of goes over the pocket a little bit, and then when the ligand comes in, it has an induced fit, where it like hugs the ligand a little bit, causing a conformational change in the whole protein. This affects the intracellular side of the protein, which can then activate G proteins.
So with this now conformational change of the receptor, we have the activation of the G protein, Katie just said. Right now, we don't really know what the exact affinity is for different proteins that the receptor activates, but we do know that it's dependent on relative levels, so just in the cell. And from studies, we found a few main proteins. The first one is going to be our alpha G protein. The alpha G protein inhibits your adenyl cyclase right over here, which Katie will talk about later. And then the beta gamma protein complex, it causes a phosphorylation cascade right on this side.
And the phosphorylation cascade, it transfers the signal through activation through all of these different kinases. And of course, a kinase is a protein that transfers a phosphate group from the cytoplasm onto another protein, in this case. And all of the proteins are kinases, meaning they take turns phosphorylating each other all the way down until we get to the extracellular receptor regulated kinase. And there are 2 of those. And the second one then enters the nucleus for the cellular response. But when we go over here, the alpha G protein, like Kyler said, goes to adenylyl cyclase. And it attaches to the adenylyl cyclase, and it stops its process, which means that the second messenger levels of cyclic AMP, which adenylyl cyclase converts ATP into, they are prevented from raising, which means that the cyclic AMP phosphorylation cascade that it starts is inhibited as well.
So at the end of that phosphorylation cascade, we have cyclic AMP responsive element binding transcription factors, or CREB transcription factors. And that is one of the 2 sides. We have our ERK and our CREB. So the ERK, it's going to activate the positive limb of the feedback loop. That's dealing with 2 main clock genes. We have our brain and muscle ARNT-like 1, otherwise known as BMAL1, and our clock gene itself, the circadian locomotive output cycles kaput gene, or just clock. So BMAL1 and clock genes, they're going to activate period 1 and cryptochrome 1 genes. The CREB transcription factors, they also act as the negative limb, and they mess around with the period 1 and cryptochrome 1 genes as well, which then will inhibit themselves as they are produced, a negative feedback loop.
And all of this stuff is shown through this very intelligently designed experiment. And in this experiment, the role that melatonin has on the gene expression of clock genes, it's assessed. And in this experiment, we have 3 different types of mice that are used. One, we have the control, which is just the wild type mouse. It has all of the receptors, all of the stuff, it's normal. Then we have another kind, which through a DNA knockout process does not have any MT1 receptors. And then the third type, through the same process, doesn't have any MT2 receptors. And after that, of course, we have the dependent variable, which are the actual expression of the PER1, the CRI1, the clock, and the BMAL1 clock protein expression. That is assessed through the corresponding mRNA levels in the cytoplasm. And as we can see from these figures here, the wild-type mouse, it's got these very normal spikes in clock gene expression, because it's normal, it's wild type. And then in the mouse without any MT1 receptors, look, the expression of the clock genes are almost gone. They're completely or almost completely diminished. And then when we add back in the MT1 receptors, even if we take away the MT2 receptors, it goes back to normal, just like that. Which means?
This all means that our melatonin ligand, it has a very specific pathway with a very specific receptor, which causes a very specific transduction pathway with a very specific response. As shown with this experiment, as Katie just said, the MT1 receptor is the receptor that causes this whole pathway. Without the MT1 receptor, we're not going to have our clock gene expression. So melatonin and the melatonin 1 receptor all together makes both the positive and negative limbs of the clock gene and therefore circadian rhythm feedback loop, which we all love. Thank you guys. Good night.
Sleep well.